So welcome everyone. Thank you for joining. My name's Keenan and I'm the manager for the data working group of the Global Alliance, which is one of the 4 working groups of the Global Alliance. The other 3 being the security working group, which is focusing on the technology aspects of genomic data sharing. The regulatory and ethics working group, focusing on the ethical, legal and social implications for the Global Alliance, and the clinical working group, working on approaches for sharing and linking clinical and genomic data. But for the rest of this talk, I'm going to be focusing on the data working group, which is led by Richard Durbin of Sanger Institute, alongside David Hauser of UCSC. And its purpose is to create international standards as data schemas, APIs, and web services for the interoperable sharing of genomics data worldwide. And its goal is to enable DNA data providers and consumers to better share information and to work together on a global scale, advancing genome research and clinical application. And why do we need this? So as all of us here know, we're in the year of the $1,000 genome. The costs of sequencing have plummeted and sequencing technology is proliferating. And global sequence reduction is doubling every 18 months, as evidenced by the rate of deposits at the ENA. And to date, it's been estimated that more than 100,000 genomes have been sequenced globally, but mostly as whole exome. But by 2025, it's projected that we'll sequence over 100 million whole genomes cumulatively. So we need this because genomic data is being produced on a massive scale worldwide and will continue to grow. And the problem is that data has currently been and historically been organized in silos by type, silos by disease, silos by institution, and silos by country. And the data working group was formed to specifically address the technical barriers of the data sharing, the other working groups focusing on the other aspects. Because if action is not taken now, then we risk 100 million genomes fragmented into silos across the world in the same way as health records are now in many countries, where the data is there, but it's just not available and not shareable. So we want to break the silos to create a new era in standards for the exchange of genomic data. So the data working group is comprised of around 250 active members, about 400 in total across our mailing lists. And there's a 60-40 research institute company split, and members include EBI, Sanger, Elixir, UCSC, the Broad, NCBI, Genomics England, and more. And on the commercial side, we have Microsoft, Illumina, Google, Amazon, 23, and others. And the work spread across 10 task teams, each with around 20 to 30 members across 12 countries. And the data working group composes at least 50% of the Global Alliance, and that's being kind to the rest of the Global Alliance and saying that. So we've been organized into teams creating APIs. And our work spread across 10 task teams, each with about 20 to 30 members, and each team addressing a particular piece of the genomic data puzzle. So we have groups working on genotype to phenotype models, RNA seq expression, variant annotation, sequence annotation, reference sequences, reads, variants, metadata. And broadly, the primary areas are the raw reads and assembled reference, the raw reads, and the secondary areas addressing annotation and metadata analysis. So this slide is an alternative view of the previous diagram, and a more astute view may have counted only 8 teams in the previous slide. The other 2 are the benchmarking team, working on to benchmark variant calling, and the file formats team, which underpins everything that we do. And up here we have the reference server that I'll get to in a few slides. So the Global Alliance file formats team are the official custodians of the file formats used in genomics worldwide, and that's the BAM, CRAM, VCF files that we're all so familiar with. And the file formats team is moving with us to an era of binary streaming web services, and moving away from passing flat files around from location to location to silo to silo. So if you're a user and you want to share genomic data via our data models and APIs, you can go about it in 2 ways. The first is you can take our schemas here, or a part of them, that you're interested in, and you can go off and implement and code them up all by yourself. And this is the approach that's been taken by this list here: the EBI, Ensemble, Microsoft, Santa Cruz, UCSC, Google, and they've all implemented parts of the API. There's no group that's implemented it in its entirety. So, for example, Ensemble are observing the variants and the variant annotations via their REST server. Google are focusing on the reads of reference sequences and some annotation. If you've used Google Genomics or are familiar with Google Genomics, this is their implementation of the Global Alliance API or subcomponents of it. This is the Ensemble an endpoint on the Ensemble REST server, implementing the search variants on the REST server. So if you've needed the time and inclination of the resources to implement to go and interpret a technical spec and schemas, then you're in luck. Because the second way to explore data models is to download and run our reference implementation of our APIs. So it is a server that you download, place some BAM files or a VCF file in a directory, and you just start it up, and you have our REST server, web server running, serving our APIs. It's written in Python. You install from the command line using standard Python tools, and you can go and read about it, look at the code, contribute to the code on our GitHub on this URL. There'll be a number of URLs through this talk that you might find useful, and these slides will be shared afterwards if you want to come back and follow up on any of this. So these are the main components of our reference implementation offering. We have extensive documentation. We have our schemas with the interoperable API. We have a compliance suite, which is a verification tool for all of our implementers. And we have the reference server, which is a validation of our APIs and which is deployable on existing data sets and traditional file formats. It's a diagram showing roughly the server sits in the data store and clients can connect from a browser. We have IPython notebooks, so you can write your own custom code. If you want to read more about any of this, you can visit GA4GH.org, and there you can read about our task teams, read our documentation, links to our GitHub, read our docs, test out the server, the reference server. This is a reference server which is running on 1kgenomes.ga4gh.org. Again, these slides will be circulated afterwards. And that's the 1,000 genomes data set, 2,500 BAM files, about 30 gigs each, 75 terabytes of data, and this associated VCFs and GRCH37. And you can go there and play with the server responses in API. This is our GitHub site. This is our preferred mode channel of interaction with us. We like that people to visit, make pull requests, create issues, let us know how they'd like us to move forward with them. And again, GitHub.org, GA4GH. So I've just given you a lightning run through of our APIs, and taking a moment to pause. So we've been working for about 2 years producing all of these tools and the data models, the different types of genomic data that we want to share, and we're just a nudge away from declaring final victory on the APIs this month. In fact, we created the bricks, and now we want to take what we created and build a house. Now we want use cases to set the direction. We want to move away from our data modeling activities and direct our energies now towards projects and use cases. We want to eat our own dog food. We want to use our own APIs in the projects that I'm going to describe in the coming slides. The data working group is now moving away from data modeling and creating APIs to now taking a focus on projects and use cases. The first of these is the BRCA project and the ACGI Cancer Gene Trust. The goals of both of these projects is to build a mechanism for publicly sharing cancer data mutations and clinical data. This is the BRCA Exchange website. This taking a focus on BRCA variants, and this is the Cancer Gene Trust. That's a project to collect DNA mutations and clinical data from at least 1 million of the 14 million new cancer cases each year from across the world, and to provide a mechanism so qualified researchers can dig deeper into restricted access data. You can read about this further on our GA4GH.org website. Second project is a collaboration between Ensemble and UCSC Browser to provide Global Alliance web services. You've already seen that Ensemble are already providing some endpoints via rest.ensemble.org. UCSC are going to take the reference server and make it as a REST interface to the UCSC browser genome gateway. At the end of this project, we'll have both UCSC and Ensemble serving data using the Global Alliance APIs. Further project, we have a collaboration between Global Alliance, FHIR, HL7 FHIR, ClinGen, and Genomics England to create a common variant model that all can use. We have the streaming API, project led by Ewan Burney here at EBI, and that's a high-performance binary streaming server to serve BAM, CRAM, VCF, and with optional transcoding on the fly and slicing across multiple BAM files to serve slices of genomic regions as a single binary stream. We have 4 prototypes, one at EBI sitting on top of EMA, archive-wide, Google, Sanger, and DNA Nexus, and that's moving along nicely. Then we have our data discovery projects that I'll get into in a few slides. But before that, I'd like to wrap up the data working group section of the presentation, showing this is where the data working group sits in all of this activity. We're providing the software, API, tools, and personnel to the projects and to enable them to fulfill their mission. And down here, we have Pekin, which has become a focal point and for the activities of many of our working groups and projects, and provides an exemplary use case for the activities of the other working groups. And now I'm going to take a deeper dive of Pekin, especially here, given its role as a flagship of Elixir. So Pekin is a web service that can be queried for the existence of information about alleles of interest, and it's designed to be simple and easy to understand, and can be implemented on top of any genomics data store. And the user interacts and queries a Pekin in a very simple way. They simply ask, do you have any information in any data set that you hold about this allele at this position? And the Pekin responds simply yes or no, but may optionally include metadata about the allele. This is an example query. The user or web browser issues a query in this syntax, coordinate-based syntax with the allele, and the Pekin responds simply true or false. This is the response. Very simple. It's optional metadata response, and here it's returning the frequency of that allele in the data sets that it holds. So Pekins are designed to be very simple, very technically simple to implement, but even with this simplicity, Pekins still provide utility to researchers. And they do this in the context of how researchers currently gain access to restricted data sets held in institutes behind data access committees. The researcher must first make an educated guess about whether a data set might be useful to them, to the research. And they make an application to the DAC. The DAC meets, makes a decision. If they grant access, the user then must download the full data set, which may be a large volume of data. It may take a long time. The process might take several months, start to finish, between application to final download. And at the end of all this, data set may not be useful to the researcher. And this is where a Pekin can help. The Pekin sits at the interface between the researcher and the DAC. The researcher can ask the Pekin, do you have any data sets for this allele or these alleles of interest? The Pekin queries the data set on the user's behalf, responds yes or no, and on the basis of that response, the researcher can then decide whether to embark on the DAC application process, potentially saving months of time. Pekin's been hugely successful from its inception. We've had great adoption, and these are just some of the organizations who have lit Pekin. We've got over 60 Pekins now lit, sitting on top of over 200 data sets and representing over 100,000 individuals within those data sets. So we quickly realized in the early days of Pekin that we'd need a way to search across beacons, federated beacons with no central data store in a similar model as the internet. So we created a directly in search engine for public beacons. And this allows searchers to quickly discover who's seen a genetic variant of interest from within a growing network. And it can be visited at beaconnetwork.org. But here's some screenshots. This is what it looks like. You visit, you enter your query here. Beacon Network will query all of the beacons that it knows of and respond for each of those. Yes, no, found for those. This is an example query with some metadata coming back from the Broca exchange that I mentioned earlier. So the initial experiment has worked. The beacon is, people have proved that they're willing and able to share data via beacons. We want to move beyond that simple yes-no response. We want to enable richer queries and exchange of more complex data sets. And we want to leverage this Global Alliance Data Working Group APIs. So instead of a user issuing a query, the beacon responding yes, no, and the user applying for the DAC, we want to allow access to the data directly via the APIs instead of downloading the files. So once you start sharing more complex data, then that comes with more complex privacy and ethical concerns. You can't just serve full genomic data via anonymous requests as we're doing now. So we've created a tiered data access model whereby data of different sensitivities can be shared via beacons according to the levels of assurance and authorization of the user requesting it. And these are the three tiered access models. We have the public, registered, and controlled. The public access is where most beacons are now, anonymous queries from a user issuing a simple yes/no response. And we have the registered access level whereby a user may identify themselves with an account and declare themselves and be identified as a bona fide researcher. And through this level, for example, you might serve allele frequencies. And then we have the full controlled access level where a user has been identified, authorized, and signed an agreement or agreed to data use conditions. And through this level, a controlled access level, might serve down to individual genotypes. The beacon operators may choose which levels of access to support. They may choose to support all, any, or none. And it's up to them. They have complete flexibility in what they want to do as part of this tiered access model. This is an example of some policies of some beacons currently live. We have the Harvard Personal Genome Project, which is a completely open access model. And their beacon, they provide from allele existence, which is the yes/no response, right down to the individual phenotypes and the individual genetic data, all by anonymous responses. dbGaP, on the other hand, has taken the allele existence, the yes/no, by anonymous request. Then, if you wish to discover frequency by ethnicity, you must be registered, for example, a bona fide researcher. You must have signed a user agreement and be completely authenticated to get access down to the individual genetic data and individual phenotypes through dbGaP. This is an example architecture of a beacon. You have the API sitting on top of the data store. But to move to this tiered access model, we need an additional layer. We need this authentication authorization, and this is where Elixir have been collaborating most closely and providing and really driving Beacon forward. So Elixir GA4GH Beacon project is a flagship project of Elixir with three objectives. Three objectives, and by the end of 2016, the aim is to have beacons laid in several Elixir nodes, and to provide an Elixir reference implementation of Beacon with those three authorization levels. And the Elixir Beacon will be integrated into Elixir AII, the authentication authorization infrastructure framework. And an Elixir identity should give access, for example, to all aggregate data within Elixir services through a registered access layer, and that's given that user also accepts the terms and conditions and belongs to that bona fide researcher group. By end of 2016, beacons in several nodes. The Elixir Beacon project will continue in 2017 and will be compliant with the Global Alliance Beacon Project standards and security working group policies. Some milestones for the Elixir Beacon. They're going to leverage the EGA beacon that's already existing, pushing the Global Alliance specification. The Elixir Beacon is version 0.3 of the spec, going to include those data use conditions, and the first iteration is available on this GitHub link. You can follow up the slides afterwards. So Beacon has been the focus of a hugely successful partnership with Elixir, and that culminated recently at the end of June with a full two-day meeting between Beacon, the security working group, and Elixir held here on campus in Hingston. And now we've got some extremely useful new direction and use cases and action items going forward from that meeting. Just to wrap up Beacon, so if you want to be involved, you can write a Beacon, use the Elixir reference implementation, you can join team or mailing list, you can use beacons and contact me if you'd like to become involved. So I've just presented on the data working group the APIs and Beacon. And just to remind you, the goal of the data working group is to promulgate standards to enable the discovery and sharing of genomic data via an ecosystem of interoperable web services. And through our APIs and standards, our projects and working groups, we want to tip the balance in global genomic data sharing. And we want to break the silos and create a new era of standards for the exchange of genomic data. That's the end. Thank you very much for listening and joining.